machine learning, so that's going to be pretty cool. Today we're just going to wrap up our discussion of BioPython, talking about this sort of other side of its functionality, which is dealing with molecular structures. So this is really like a really important aspect of dealing with biological data, at the very least, molecular biology, as far as that's concerned. So last time we looked at sequences a lot, right? So how can we deal with information regarding sequences, and how do we store them? How do we perform these convenient functions on sequences? And basically, sequences can be DNA, RNA, and protein. And pretty much, all of the function of a molecule we know is encoded in that sequence somehow, okay? So if we know the sequence, we kind of have all the information we need to know everything about that molecule to a degree. So the screen is purple, nothing I can do about that. But the notebook is on the course webpage, so you guys can follow along there. I'm sorry about that. We're going to have to call the service people. Okay, so the sequence gives us a lot of information, but at the end of the day, inside your cell, molecules kind of have these geometries and shapes. That's actually what determines how they're going to function, okay? So I pulled out the sequence, for example, for this protein called a dopamine transporter protein, which is responsible for taking dopamine out of your synapses and making you kind of stop being happy all the time. So all these drugs that, make you feel less depressed or happier interfere with this kind of protein so that you're always having dopamine in your synapses. So I thought it was a pretty interesting example. So this is its sequence, right? These are just a bunch of amino acid letters, and it's pretty much just a string, right? So BioPython lets us work with that in the sequence sequence library. Now we're going to look at what this actually looks like in 3 dimensions, okay? And hopefully we'll get a better idea of, how this molecule actually performs its function. So there's a very big database where scientists basically put data associated with the structure of a molecule. It's called the Protein Databank. I have a link to it here. Basically, it's a big website where a bunch of interesting proteins and RNA and DNA structures are stored. Okay, so whenever scientists kind of go in and figure out what a molecule looks like in 3D, they'll put it on this website. Okay, and you can download the data for free. So this is that dopamine receptor. All right, we can look at it carefully. Right, we see. I think around here there should be a dopamine molecule. I think it's this one. Anyway. So we're going to be using this database just to play around with some of the functionality here. So there's a lot of information that the sequence right away doesn't tell us, right? This whole kind of 3D orientation is really what's driving the function of this molecule. So we want to be able to manipulate that data and do something with it. So what is a structure? Basically, a structure, when I say structure, I just mean a 3-dimensional sort of vector or 3 numbers describing every atom in a molecule, okay? So proteins, DNA, and RNA are all composed of atoms, and in your body or in a cell, these atoms have a specific, location in 3D space, and that's what defines the shape of a molecule. And that shape tells us gives us a lot of insight on the function of that molecule. Just some aside, often the technique that's used to obtain these, you guys might know, is called X-ray crystallography. So, these chemists and biologists isolate a protein or whatever in the lab, and they shoot some lasers at it, and the way the electrons from the laser diffract tells them, how these atoms of the molecule are placed in space. Okay, so this is the X-ray diffraction pattern of the DNA double helix that was really famous. We don't have to worry about how to interpret that or really how it works. This is just one of the techniques that's widely used in order to obtain the type of data that's found in this database, for example. Okay. So now I'm going to go into really the BioPython side of things, how we can deal with these pieces of data. So we did biopython.seq last time. This time we're doing biopython.pdb for Protein Databank. Okay. So biopython.pdb has all these useful functions that let us deal with structures. All right. The main format for 3D structures is either PDB, it's known as PDB, or MMSIF, which stands for crystallography information file, something like that. Okay. So when we want to use any of these BioPython functions, all we have to do is import them. So we say from bio.pdb import star. So I'm just going to import all of the names that were defined in that module onto my local namespace, my global namespace, and now I can use all these functions. Okay. So the first thing we're going to do is actually get the 3D structure of this dopamine transporter, and we can do it without having to touch the browser or anything, just by saying pdb list, right? So this object kind of has a list of all the 3D structures that are on that database, and then we can say, okay, that attribute that's going to have an attribute called retrieve pdb file, and it'll go on the web server and take out a specific protein file or structure file with some specific ID that we give it. So this dopamine receptor that we're interested in is called 4xp1. So every entry in this database has a code associated to it. It's 4 characters long. So this one's 4xp1. Right? In order to find this, I really just typed in here like dopamine or whatever. So there's tons of examples. So obviously, all right, we want to retrieve this file from that database. So you need to be connected to the internet, obviously, to do this. But it does it. And it says, okay, the structure already exists, meaning I've previously downloaded this file. Not a big deal. And then it gives you a warning saying the default format has changed from PDB to MMSIF, basically. So we're going to be working with this new format called MMSIF. All right. So yeah, that's it. So we've downloaded this file. It should be in our local disk now. In order to check that, let's say import OS, that module I told you guys about. It lets us list all the files that are in our current directory. Okay. So we see here that I have 4xp1.sif, and I have this folder called xp, which is actually where it went first. If I just list that directory, I'll see 4xp1.sif. So it downloads into this folder. It creates a folder for you and then puts it in there. Just for convenience, I copied it onto my local drive, my local folder, but it doesn't really matter. So now we have this file. If we were to open it, we would get something like this. Okay, it's huge, bunch of entries, lots of data. It would be a huge pain to actually write manual code to parse all this. Thankfully, BioPython has a parser. So all we have to say is, all right, we're going to create a parser object by saying mmsif parser, okay, and giving that a name. So let's run that. And now we have this object that can parse files that are in this specific format, okay? So we can say, now that we've created this object, we can say parser.getStructure has this attribute, getStructure. We can give the structure a name, just a string that we specify. So we'll just use the same name as is on the database. And then we specify where that file is located, okay? So the BioPython call created this XP directory and then stored the file here, all right? So we just tell BioPython that go there, look for that file, and extract all the information, okay? So that should work. Takes a bit of time, all right? It gives us some warnings saying chain A is discontinuous at line 7477, whatever. So these files are not very perfect. There's often missing parts or holes or something like that. So this parser kind of lets you know about that, but it still works. It doesn't crash. This is just a warning saying, look, this has some funny parts to it, but you don't have to worry about it too much, and we'll ignore it. But yeah, that's something to know. A lot of these experiments kind of aren't perfect, or sometimes there's mistakes in how they enter the data. So the parser recognizes that and says, look, maybe there's some missing parts to your structure, but it's not too severe. All right. So now we have this structure object. Okay. So this parser.getStructure call returns an object of type structure, which has all the information that was in this file here that describes, basically the X, Y, and Z coordinates of every atom in this protein, and some other extra information. Okay. I want to look at the attributes of this object. So we can do that right here. All I did is I defined a function that calls the dir function on an object, which is the one that lists all the attributes of an object. And I'm just getting rid of the ones that have 2 underscores, which are not super interesting for now. So that's just this function I created called cleandir. Okay. So now whenever I call it, I give it an object, and it'll give me all of its attributes, except for the ones that have underscores in the beginning. Okay. So we see there's a bunch of stuff here that we can use in this structure object, and we'll get into that and look at a few instances of it. Okay. Is this clear for now? Okay. So BioPython organizes all the data in this file in this sort of hierarchy. We're not going to look at all of it, just kind of the core elements of it. So it says, okay, we have the sort of the most general thing, which is just a structure, right? Just one big blob of atoms, okay? And inside that, you can have several models, okay? So sometimes you'll see in the database, not in this example, but you'll see multiple copies of the same protein. So the people isolated multiple copies and put them all in the same folder, in the same entry. So Python recognizes that and separates them into different models, okay? So you know that one model is sort of like one unique occurrence of that protein, all right? So for now, we only see one protein here, so we're going to say we only have one model. That should be the case. And then inside that model, you have what's called a chain, all right? Or multiple chains. And chains are really just literally chains, okay? So if you think of a protein, right, it's kind of a strand of amino acids, right? Chain, they're just all linked together, and they have some 3D structure, right? That's this like blob that you see. And sometimes you can have multiple sort of chains interacting together. So you can see kind of like, here's one blob, that's probably one strand of protein kind of in one location. Then here's another one in the middle, and then here's a bigger one, all right? So these are kind of three different parts of the protein, right? Sort of continuous amino acid chains. So we should have three of those in our structure, okay? And then each chain then is made up of residues, right? So just amino acids. In the case of protein, in the case of DNA, it would be bases like A, C, T, G. But here we have amino acids, okay? So every chain is made up of amino acids, right? It's just a sequence of amino acids. And each one of those amino acids is then made up of atoms, individual atoms like carbon, hydrogen, nitrogen, okay? Is that clear? So we're going to be able to access after having parsed the file, we're going to be able to sort of access this structure at all of those levels of analysis, okay? So sort of the most fundamental thing is the atom, right? From that atom, you can sort of abstract your way up, right? Okay. So that's just, we're going to focus on that core, right? You have models, then you have individual chains, then chains are made up of residues, and residues are made up of atoms, right? A residue is just another word for an amino acid or a nucleotide, all right? So there's a really nice tool that I just found out about that lets us actually look at these 3D visualizations of structures in the notebooks. So I went ahead and installed it. I put here the instructions for you to install them if you wanted to try it out. I found it's pretty handy. It's quite nice. What people usually use to do sort of more involved things is tools like Chimera and PyMol. If you're interested in this stuff, they're super widely used and they're used for visualizing structures in 3 dimensions. Okay, but here we just need to sort of quickly look at it. So we're going to do it directly in our notebook. So I'm using this module called ngl-view. And so I'll worry too much about how that works, but basically I can give it a structure object from BioPython and it'll display it for me in the notebook, okay? So, if I run this, here we go. So basically what the chemists get after doing X-ray crystallography is basically something like this, okay? They get a big cloud of atoms, right? Every sort of ball here is an atom, right? And they're joined by sticks, which are bonds, okay? Right, so this is essentially a protein, right? And then obviously it's kind of a messy way to look at things, so we can kind of get a cartoon visualization of that, which is called a ribbon representation. So instead of drawing individual atoms, you just say, okay, look, every strand of protein is just going to be like a ribbon, and you can sort of more clearly get the idea of what this thing looks like in 3 dimensions, okay? And we should see here, for example, this is like, this is a sort of a small molecule that's binding to this protein, okay? I don't know if this is dopamine exactly. I think maybe this one down here is dopamine, but basically you also see anything that binds to this protein, right? So you can start to get an idea of, which parts of the protein are actually functioning in this protein's task, okay? Any questions? All right. Yeah, so this is called the ribbon representation, and this is called basically, oops, it changed it, but when you saw all those atoms, it's called ball and stick, okay? Yeah.
You said we might need a Druid extension installation, or
Sorry?
install it, how come they use Druid?
Oh, so you have to run these commands here in your terminal. You say install nglview bioconda, and then you type in this command. Yeah, and that should enable you having this. It's called a widget, so you can add sort of cool external things to your notebook. Okay. So now I'm just going to take you through parsing or sort of dealing with these structure objects. So like I said, inside of one of these data entries, you can have multiple copies of a protein. Those would be stored in separate models. But for now, we only have one. So our main thing that we got from parsing was this structure object. Okay? And if we had several models, we would be able to iterate over them. So if I said for model in structure, I should be able to print out sort of the representation of every model. And here we only get one thing. Okay, so we only have one model. So that's fine, that's nice. We only have to worry about one thing here. And now, like I said, that model then is made up of chains. So these different, I think here every color represents a chain. So sort of each continuous chain of amino acids is going to be given its own identifier. Okay. So we can iterate through all the chains in our protein if we want, right? So let's say, all right, we're working with model, with the first model, which is the only model. So we say structure at 0. That's the model of our protein. And then we can say, all right, let's see what all the chains in this protein look like. We can say, all right, since models are made up of chains, we can iterate through all the chains in a model. And I'll print the attribute of the chains, a chain called .id. So every chain has an ID attribute. So here we go. We got 3 chains. If I print out the chain directly, I get this sort of representation of the object. And I can access its attribute .id, right? And I printed it out here. So for example, this chain's ID is A, L, and H. All right. So there's 3 chains. If we go back here, we can sort of put our mouse over it and see this is the H chain. You see there's 182.H.Ca. So the .Ca stands for carbon alpha, so that's the atom. Then before that is H is the chain, and 182 is the number along that chain of that residue. Okay. For example, here we're looking at chain L, and here we're looking at chain A. All right. So we have 3 chains. Let's focus on chain A. So I'll store that in this variable here. So I'll say model at A, right? Since models are made up of chains, I can pick out a chain. So it's kind of like a dictionary. So I'll say model at A. That should be all the information associated to chain A should now be in this name. And now chains are made up of residues, right? That's how you form a chain. So I can iterate over all the residues if I wanted. So I'm going to do that. So for every residue in chain A, I'm going to print out some attributes of it. So the residue's name, that's in the attribute .resName, and its number along the sequence, that's res.id at 1. So the ID has 3 pieces of information, so it's a tuple, but I'm only going to show you one, which is sort of the number along the chain, right? So if you look at these chains, along these chains, every amino acid has its own number, right? And this should be sort of increasing, right? So here we're at 205. Oh, it's going to jump around. 122, 121, 120, 119. Okay? So along this chain, we have amino acids sort of holding hands, and they each have a number. All right, so we can just print that. All right, so we've got a bunch of residues here. So this is an aspartate. Its number is 25, right? We can go on all these residues, making up chain A. All right? So now we can programmatically access all the residues in a PDB file, right? So finally, residues are made up of atoms, okay? And at the end of the day, that's sort of the indivisible object here that we're working with. So let's focus on one residue here. Again, so if a chain is made up of residues, then we can access an individual residue given a chain. So given chain A, we can say, all right, give me residue 56, right? Just like a dictionary. Store that in there, and I can print it out. And so I have this residue object here. All right, it's leucine. Its sequence, its position in the sequence is 56, and we don't have to worry about these 2 other attributes. Okay. So finally, residues are made up of atoms. All right, so I can do the same thing, iterate over every atom in this residue. And I'm just going to print out the name of each atom. So for every atom in this residue, I'm going to say atom.name. All right, so I'm going to get a big list. Oh, no, not big list, because there's just one residue. So this leucine residue here has a nitrogen, carbon, carbon alpha, carbon, oxygen, carbon beta, and so on. Okay? Is this clear? So we've just kind of broken down this structure all the way down into its individual atoms. All right? If I wanted to put this all together in one big messy for loop, I would say, all right, if a structure is made up of several models, I can iterate over every model. Then for every model, I can iterate over every one of its chains. For every chain, I can iterate over every one of its residues. For every residue, I can look at every one of its atoms, and then I can print that atom, okay? And when you hit enter, it's going to be a huge list, right? Boom, okay? So we have every single atom in here. All right? So that's basically it, okay? There's a lot more about this, but this is kind of what's going to get you started, okay? And now we're going to sort of go down to the atomic level and do a little mini project, okay? So we want to, let's say, we're trying to design a drug here that makes people super happy. So we're going to need it to bind where that dopamine is binding, so that it's no longer able to clean up all the dopamine from your synapses. So you're going to be permanently happy. Okay. I think this is sort of what cocaine does. So let's say we wanted to make up our own cocaine here. We would need to know like what part of the protein to target, right? We want to create a chemical that's going to sort of get in there right where the dopamine would bind. So we need to know what this dopamine is binding to, all right? So in order to do this, basically we want to pick out residues that are defining what's called a binding pocket. So all these amino acids that are the ones that receive that molecule that we're interested in, which is dopamine. And if we know what it's binding to, then we can, if we know some chemistry, say, okay, well, this type of drug is going to like to bind to those residues, okay? So we would be wanting to programmatically pick out something like, I don't know, let's say we have this, it's not very clear here, but you'll see if you run. Okay, so here's a ligand. So a ligand is just a small chemical, like a drug. And we'll say, okay, well, it kind of likes to bind to this residue here and this residue here, right? So just pick out everything that's kind of around it, and then later on we'll be able to analyze it, okay? Obviously, like you can do this right now, right? You could look visually at this chemical and say, okay, well, it's binding here to a leucine, then an alanine, then a isoleucine, and so on, and then you're done. But this is a little bit low tech. Like, what if I told you I'd automatically give me all the binding pockets of all the ligands in the PDB database, all right? We want to be able to do this automatically and programmatically. So if we do it using BioPython, we'll be able to do that really quick, okay? And we're going to do that in two ways, all right? And one way is going to sort of teach us a little bit about how these messy files are structured here. These are called Emissive files, all right? And the other way is sort of directly working with the atoms in BioPython, okay? So Emissive files are basically like dictionaries, okay? They have keys and values. And there's tons and tons of entries in them, or keys, that when chemists or biologists isolate the 3D structure of a molecule, they can add that information into that file for someone else who's interested in looking at it. So they'll often annotate it. So say if they know that there's some sort of binding happening, they'll say, all right, there's a binding site in this protein at this location, and these are the residues that are involved. So in a way, they've done a lot of work for us, but we're not going to trust them, and we're going to try to do it ourselves as well. But this will help you sort of understand how this file works a little bit. All right? So basically, there's tons and tons of different possible entries in this file. But we're interested in things that are non-polymers, okay? Non-polymer here means something that's not protein, DNA, or RNA, all right? So it's some chemical, like dopamine or cocaine or whatever. So the researchers went and said, all right, I'm going to label anything that's not a polymer, non-poly, and I'm going to give you a bunch of information about it, all right? And basically, this is what a typical MMSIF entry looks like. It's a little confusing, but this is how it works, all right? Every one of these rows with an underscore at the beginning is sort of the label of a column, okay? So this says asim ID would be the label of this column, the first column, okay? The second underscore entry here would be the label of the second column, and so on. So there are 10 labels here and 10 columns down here, okay? So that just tells you what every column of this sort of table means, all right? So for example, the last label here, PDB insertion code, would be this last column here, all right? You don't have to worry about actually extracting this yourself. BioPython does it for us. But basically, that's how it works, okay? So basically, you would get a dictionary, right, where each key is one of these labels, and the value of that key would be a column, okay? So this column, the first column would correspond to the key here, asymmetric ID, right? So the way you know what these means is you really go look at the documentation, and they'll map out what every one of these entries means, and then you go into the file and you look for what you're interested in, right? So what I did is I did that, and I found, okay, non-poly means, these are all the ligands, okay? So for example, we have a sodium, we have a chlorine, we have this thing called MAL, I don't know what that is, NAG, I don't know what that is, LDP is the dopamine, okay? So it's saying that there's a dopamine, this LDP, that's dopamine, and it's found in chain A, all right? And it has a residue ID of 708, and its residue name is LDP, okay? That's what these weird cryptic things mean. You just have to Google it. So another thing they label is the binding sites, but we'll look at that later. Okay, so let's actually look at that in Python. So the way you extract this information is you use a module called mmsif to dict. Okay, so you're turning this mmsif file to a dictionary, right? And you're giving it the path to your file, all right? And then that just automatically creates a dictionary for you. I'm going to print out all the keys in the dictionary, all right? This is all of these weird headers that we saw are all the keys to this dictionary, all right? There's tons of them. We know which one we're interested in, so we'll just go for that. I found another one that was interesting called struct site. So this actually stands for binding sites on your protein, okay? So they've actually gone and labeled all of the places they found chemicals bound to the protein, so you can actually automatically go and look for them. So again, these are just column labels, and these are the actual entries. So this is column 1, column 2, column 3. So the first thing is the struct site's ID. So each one of them gets a specific ID. And then there's, for example, the last column means details, so a little string describing this binding site. So if we looked at the last column, I think, which one had the binding site? So binding site for LDP, right? So this is the dopamine, right? So its ID is AC7. These are the details associated to it. So it's binding site for residue IDP at chain A, residue ID 7. Okay? So now we know that there's a binding site that has an ID AC7, and it binds LDP, right? And this is its residue ID. So we could go into our little for loop over all the residues and actually pick out, this chemical that's bound, all right? So let me just show you, for example, one entry in this dictionary, right? Like I told you, these strings mean titles for columns, okay? And I zoomed in too much here, but basically this is one row, okay? Here's one row. The details column is here, right? The last header on my list of headers here is details. So it should give me the details to all these binding sites. And here you go, we have a list of the details of all the binding sites, okay? So now we can look at them and say, all right, we're interested in LDP. So we know that there's a binding site here associated to LDP, right? Now there's another entry in this file that actually tells you what residues are in the binding site, all right? And since we know the ID that we're interested in, AC7, right, the researchers went and said, all right, we're going to give a name to this binding site for LDP, and we're going to call it AC7, right? Right here, AC7, binding site for residue LDP. Then we can give you more information on that. So again, we have a bunch of keys, and each key is going to be mapped to a list of values, right? And this basically says, for example, this binding site AC1, right, is going to have this alanine, this asparagine, serine, asparagine, H2O, water. Okay, so it's just a list of residues that are associated to a specific binding site. Okay. And there's a lot of other information. For example, citation.title tells you the title of the paper that produced this crystal structure. So neurotransmitter and psychostimulant recognition by the dopamine transporter. Okay. So there's a website that gives you the details of all of these types of entries. You just have to browse through it, and eventually you'll get what you want. Okay? So I wrote out the code to pull out all the residues in the AC7 binding site. All right? So what I did was I got all of these dictionary entries, right? So this big dictionary that has all the information for our structure. Every key is one of these columns, right, that we saw. So for example, the site ID, I'm storing it here. The list of all the site IDs. The list of all the chain IDs are here. The positions of all the residues. And I forget what this one is. The names of the residues. Okay? So these are all just lists, right, on a table. Like this, okay? Basically, I'm reading this in. So if I were to print one of these, it would look something like this, right? Just a list of values along a column. All right? And then basically I'm just iterating over all those lists in a big zip. So I'm looking over all the lists at the same position one at a time, okay? So that's what zip lets you do. You can loop over multiple lists. And because we know that they're all just rows, I can say, all right, the first iteration of my loop is going to be the first row, second iteration is going to be the second row, third, and fourth, and so on, okay? And then I can just run this. So I pull out every row, I get the binding site ID, the chain, the residue number, and the residue name, okay? So these are all the residues that the researcher said bind to this chemical, okay? So I only printed if I saw that the binding site had the ID AC7, which we saw was associated to dopamine. All right? So now I have a list of all the residues that these researchers said are in the binding pocket. All right? Any questions here? It's a little bit of a funny way to organize the file, but if you just take a minute to actually look at it and run one of these dictionary calls yourself, you'll see it's not that bad. Okay. So there we go. That's what came pre-made for us when we downloaded this file. Researchers said AC7 means binding to dopamine, and these are all the residues, their numbers, and the chain they belong to that bind to that chemical. Now let's say we don't trust that annotation and we want to figure it out ourselves. All right? Well, we can do that too. So, like I said, we can loop over all the residues in our structure. Right? Now we're going back to that structure file that we were looping over before, structure object. So remember, I got a structure which has a bunch of models. We only have one model, so we say, okay, model structure at 0. And then I use this useful attribute here, .get residues, which basically gives me a list of every residue in my protein. Okay? So what I'm trying to do here is I'm looping over every residue in my protein, and I'm saying if one of these residues has the name LDP, which stands for dopamine, that would mean I would have the attribute residue.resName LDP. Then I know that I found my LDP residue, okay? So then I just store it here. I say LDP equals this residue, and then I could break my loop, okay? I'd be done. Okay? So I printed it. So it found that a residue whose name is LDP. And now I have this residue object here, okay? It's at position 708 on chain A. Right. So now we'll do a little bit of math. So now we have our residue. Okay, so that's this sort of reference point. If we go back to our visualization here, right? Let's say I think it's this one actually. If we look here, LDP. That's right there. Okay. So this is the residue that we've selected, that we've isolated. And now we want to say, okay, what other residues are close enough to it that we're going to say that they're associated to binding this chemical, right? So what we're going to do is we're going to say, all right, we're going to make a distance cutoff. Let's say, I don't know, here things are measured in angstroms. So we're going to say, we're going to take a sphere around this chemical and say anything that's physically near it is going to be a binding residue, okay? So that's what we're going to try to do. And we can do that because every atom in this file has x, y, and z coordinates, okay? So it's really just doing a little bit of math on these coordinates. Right? The way we do that, for example, let's say, all right, so let's say we have our structure object. At 0, that gives us the model. At A, that gives us chain A. At 56, that gives us residue 56. And at CA gives us carbon alpha of that residue, okay? So atom, residue, chain, model, okay? So we can select one atom individually in one shot doing something like this. That atom is going to have an attribute .coord, C-O-O-R-D, which stands for coordinates, okay? If I print that out, I get a vector, right, of x, y, and z coordinates for this atom, okay, this carbon alpha atom. Every residue in a protein has a carbon alpha atom, so often it's used for reference when measuring distances, okay? Now let's say we want to take another atom here, residue 2. Let's say we want residue 327 on chain A with name carbon alpha. This one should also have some coordinates. Whoops. Res2, oh yeah, underscore CA. Okay, this one also has some coordinates, right? And now, because we have two coordinates in space, we can measure how far apart they are, okay? And that's quite simple. We just subtract them, right? So we can take the coordinates of the first residue, subtract them with the coordinates of the second residue, and we'll get another vector that's the difference between them. So along every axis, how far they are from each other, okay? Is that clear? Just really subtracting coordinates. All right. If you wanted to have an actual distance measure, we would want to have positive values because distance is a positive thing. You can't have negative distance, so we would say, all right, let's say we got this vector here, that's the difference between these two atoms. We can square it so it becomes positive. So we just multiply it by itself. And then to get back to the original scale, we just take the square root after, and that should give us sort of a distance vector that's positive and has the right units, right? So this is what we're going to use then to get all the residues in a binding pocket. So here's the code to do that. All right. Well, let's walk through it really quickly. So let's say we have a cutoff of 10 angstroms, okay? Which is equivalent to 1 nanometer, which is 10 to the minus 9, I think, meters, something like that. Okay. So this is our cutoff, right? We can set it to whatever we want. We can make it really small or make it really big. Obviously, the bigger we make it, the more residues we're going to consider in this binding pocket. The smaller we make it, the more restrictive we're going to be. And we're going to start off this list where we're going to stick in all the residues that are close enough to our dopamine. All right. So now all we have to do is loop over every residue in our protein and check whether it's close enough to the dopamine residue. Okay. So same thing. We say for residue in structure at 0, so that's that model. The first and only model, and then we say .get residues. That gives us a list of all the residues in the protein. And then we say, all right, if the residue happens to be LDP, then we just skip it. We don't want to get the distance to the chemical itself, so we just continue. And we also skip any other non-protein atoms, okay? And basically, you can tell which residues are not protein if they have an ID that starts with H. Basically, it's a little hack here. So every residue has an attribute .id. I'll show it to you here. So we have a residue here. Okay, so let's say res2ca.id. Okay. Ca. Oh wait, that's weird. Is it atom? Oh yeah, because this is an atom. Sorry. So let's get residue. Let's just copy this and only go up to the residue. So this will give us an actual residue. And then we say, now we print residue.id. Okay, So we got this tuple here. That's basically what describes this residue, okay? The second entry is its position along the chain, and there's this empty field here. If this is empty, then it's actually belongs to a protein. But if it's not, it'll have an H, which stands for heteroatom, which means it's not an amino acid, it's something else. So that's what I'm doing here. I'm saying if that has an H in there, that means it's not part of the protein, so we don't care about it. So I'm skipping it, okay? So if my ID at 0 starts with H, skip it. Otherwise, I actually do need to measure between that residue and my ligand, okay? So I have my residue that I'm looping over. I want to get its carbon alpha atom, right? Because distances have to be measured between atoms. So I say residue at carbon alpha, that's going to be the alpha carbon, okay? And now I have a ligand that's made up of many atoms, okay? So because ligands and chemicals often have different compositions, they don't all have a carbon alpha atom, for example. What I'm going to do is I'm going to measure the distance from the alpha carbon of this residue to every atom in the dopamine, okay? And if there's one distance that's within the cutoff, then I'll say that that's close enough, okay? So I have to iterate for every atom in my dopamine residue, okay? And I compute the distance from this alpha carbon to this current atom, okay? So that's what I'm doing here. I'm getting a difference vector from the alpha carbon's coordinates to this current atom in the LDP or the dopamine residue, okay? So that's what we did above. That's just subtracting two vectors. And then I just append, I square the vector to make it positive, and then I take the square root to bring it back down to normal units. And I just add it to this list of distances. So for every atom in the dopamine residue, I've measured the distance to this current alpha carbon, right, in this loop over my whole protein. All right. So now I have a list of distances. I'll take the minimum, so the shortest distance, and if the shortest distance between my molecule and the residue that I'm looking at is within the cutoff, then I'll add my current residue to this list of binding partners, okay? So that's all I'm doing. Any questions there? All right. If it doesn't make sense, just go back and look at it. It's not that complicated. But yeah, it takes a bit of time. Okay. So if this worked, I should have a list of residues that should be close enough to this dopamine molecule, okay? So I see I have a bunch here. Maybe I was a little too permissive. Maybe I want to reduce my distance cutoff to 5, say. We should have less residues now. Okay, we have a few less. Okay. So by reducing this distance cutoff, I'm kind of taking in less residues around this dopamine molecule. So now let's visualize this, okay? Again, I'm using this like a widget that you can use to visualize structures that we saw above. And I'm just coloring every residue according to whether or not it binds to this chemical, okay, if it's in the binding pocket. So if we have a residue that belongs to this list that we found, then we'll color it, for example, this color code. If not, we'll color it this other color code. Okay, this is a bit of a hack because it's not, this isn't really the way you should do it. So don't worry really about how it works, but figure out how to do it, and let's do it. So let's be a little less strict here. Let's go back to 10 angstroms cutoff, just so we can really see it. And then now we should see that all the residues that are near this chemical are colored red. Okay, and there we go. All right, so we should zoom in here. If we zoom in here, we should see, right in here we have our LDP, right? And all these red residues are the ones that were within that distance cutoff, okay? There we go. So you can show that to a chemist or whatever, and they'll see, okay, that's what we have to target. All right. And let's compare that to what we got by looking at what the scientists that created this structure file annotated, right? So I'm just taking a different list of residues here. Instead of the binding residues that we got, I'm looping over the MMSIF binding residues, the ones we just extracted from that dictionary. And we can view that. And we'll see that they're a lot more strict on what they call a binding site. Whoops. right? We only have a couple red residues in here. Okay. So there you go. I mean, that was just a one small example of many things that you can do in BioPython with structures. Obviously, you know, this was just doing this for one specific chemical, but the cool thing is that you can do this sort of automatically for, you know, the entire database or a protein with multiple ligands. You don't have to manually do things. You can really just, now that you know for loops basically and dictionaries, you can write programs that do really useful things for structural biology. Okay, that's all. Any questions? All right. So that's it for me. You'll see Chris next time and for the next couple of weeks.